Dan and the Brain Foundation for inviting me to give a talk here. I'm Robert Mills. I'm a computational biologist and microbiologist working at Precidiag. So today I want to share with you some of our work that we've done trying to synthesize the microbiome field. And I hope that later on, maybe next year, we can talk more about some of the translational studies that are ongoing at the company. So we're in Boston, Massachusetts. So first of all, I'll give you an overview of what I'm going to be talking about today, a little bit of intro on our company, then going into the background on the autism microbiome, and then going into our work reanalyzing the field, and then just finally some of the conclusions and perspectives that we can take away from the work. So Precidiag is working to essentially look at the microbiome of patients in various diseases, identify pathogenic processes that are occurring in certain subpopulations of patients, and develop precision therapeutics around those processes. So there can be proteins or metabolites that could be having causal roles in certain diseases, and we want to develop precision therapeutics and companion diagnostics to identify those patient subgroups and get them the right therapies. So we are trying to do a new modality in the microbiome field. So instead of trying to develop live biotherapeutics of particular microbes, we actually want to use a small molecule or an antibody approach to have a precision effect. And we are also interested in this because there's advantages to having this molecule be gut restricted, which we think is an exciting modality to explore. So the company is heavily invested in autism and colitis, and we're currently preclinical. So thanks to foundations like the Brain Foundation, there's been a lot of pioneering work in the autism microbiome field that I'll go over real briefly. So I think as many parents and clinicians know that there is often a comorbidity with the gastrointestinal system, which gives us a lot of reasons to look into the gut microbiome and see if there's potential differences within autism and controls. So people started looking at this a long time ago and started to find differences in particular species. And with the advent of high throughput sequencing, researchers started to find compositional differences using techniques like 16S sequencing and later on shotgun sequencing. But I think some of the most powerful studies that have been shown to date are some of the smaller studies that started to come out in around 2016 using fecal microbiota transplants. So really excitingly, fecal microbiota transplant from healthy individuals into autistic individuals has been able to show reduced symptoms in humans, and this report started coming out in smaller studies around 2016, and they're starting to get replicated in a few studies later on. And further, you can induce ASD-like symptoms in mice by transplanting fecal samples from autistic individuals into, say, germ-free mice or other mouse models and induce different symptoms that resemble autism. So some of these studies are also small, but replication studies are ongoing, which is exciting to see. So there's a lot of reasons to believe that the microbiome might be involved. Unfortunately, there are quite a few inconsistencies in the literature that I'm going to talk about. So in general, it's even controversial to say that there's a difference in the microbiome in the field. So some studies will say that there's clear differences in the microbiome, and some studies will not find those same differences. And there's discrepancies in really broad metrics and ways to analyze the ecology of the microbiome. Things like beta diversity of the entire composition of my microbiome versus someone else's, differences in alpha diversity or the ecology within a single sample, and large broad metrics like phyla compositions. And then this translates down to the very specific taxonomies also, where there's inconsistencies about the species and strains that are different within autism and control. So some studies that really highlight these differences are shown here, where we have a study from Dan 2020. It's one of the more higher powered studies coming out of China with around 200 individuals. And you can see autistic individuals in red. There's a very clear separation from the control individuals in green, where there's been other studies, such as a high-profile paper that came out in 2021, where autistic individuals were intermixed with their siblings and the unrelated controls. So I think as a company that's really dedicated to this approach, it's worthwhile to try to answer the question, what's generally going on here? So we took a meta-analysis of the field and basically looked at what data was available and reprocessed it in a standardized way. And generally what we were able to find about a year ago was 1,740 16S samples. There's some shotgun sequencing data that I'm not going to talk about today because it's not quite as large there. But these were coming from around 14 cohorts. And the largest cohort that we were able to generate was actually from citizen science data, where we looked into subjects that had sent in their samples for sequencing in the American Gut Project, and then we were able to pull out the autistic individuals and match them with proper controls that were the same age and sex and various other factors. So you can see that most of the studies are actually relatively small cohorts, under 50 individuals, with a few studies that are over 100 subjects. And this is a really emerging field. I think there's a lot of excitement around it. And actually in the past year, there's at least 7 more studies that have gone out, and these numbers are getting larger. So I think there's a lot of excitement around here, I will also note that there's around at least 60 studies other than these 14 studies that didn't quite meet our criteria for being included in the study, that didn't provide enough metadata for us to analyze it, or various other aspects. So the distribution is skewed towards males predominantly. A lot of the cohorts were generally under capturing ASD individuals under the age of 6. And the data is almost entirely from the US and China at this point. And studies typically will use unrelated controls, but a few are using sibling controls. And there is a large there's a lot of choices that you can make when you're performing these studies. One of the big ones is which region upon the 16S variable region you do your amplicon sequencing of. Typically researchers are using 2 different approaches, the B3, B4, or the V4, and we don't have much data on the other aspects. So when we reanalyzed all this data together and visualize it all together, you can see that a lot of the different studies will cluster separately from each other, indicating that there are large batch effects in the data. And we can describe why some of these batch effects are occurring, and it's probably largely due to the different sequencing methods that people will choose, things like the choosing different variable regions for your amplicon sequencing. So when we instead just break down each study individually and compare autistic individuals to their controls and try to find a statistical difference in these broad metrics, you'll see that around half of them will show a significant difference in their gut microbiome. And then when we try to compare large compositional differences, things like the phylum of the bacterial groups, there's a few consistencies that you'll find. Things like increased proteobacteria, I think, have replicated in many studies at this point. And increased actinobacteria are also emerging as pretty consistent trends in the autism microbiome. When you get into a more finer level of resolution for 16S, kind of the best we can do is genus level resolution. you can find a few quite large discrepancies between studies, especially when you look at Prevotella. So some studies will have very, very large increases of Prevotella in the gut microbiome of autistic individuals, while certain studies will show very large decreases. Bifidobacterium is one more consistent trend that we were able to find. So, that doesn't really explain why that difference is occurring. And so that's something I was really interested in trying to break down with the data that we did have available. So I checked all different factors that were publicly available for us to look at. And one of the major factors that influenced this finding of Prevotella was in that choice of variable region that authors were using, where if they're performing a V3, V4 study, you would typically find increased Prevotella, and in the V4 studies, you're actually finding decreased Prevotella. This kind of choice also affected certain groups of bacteria, like sulfur-reducing bacteria that have been talked about in a few studies as well as certain groups of proteobacteria. So now I'm going to move on to some of our work trying to build diagnostics based on just the fecal microbiome alone, basically comparing the fecal samples, microbiome profile of ASD and control individuals, and seeing whether we can detect the status of autism. So we've tried quite a few general classifiers and found that some definitely work better than others, and so we picked certain techniques to move forward with and we kind of collapsed the data on all of the different possible levels and applied machine learning to the data sets individually and in aggregate. Again, you can find some data sets where you get really astounding ability to detect autism with around 90% accuracy, where other studies are a lot harder to detect it. And generally, while you increase the resolution, you get better performance in your machine learning models. So we wanted to break it down by age, and so we basically split our data by every different age category that we could find. And what you can see here is that essentially we have this pocket in the younger samples that we get a lot stronger classification of autism, which actually suggests that certain microbial species that are probably influential in the development of autism could be there at an early stage, and that diagnostics based on this data could be relevant early on. So among some of the older individuals, our general ability to detect autism was a bit worse. So we broke it down by a lot of different other study factors and found that things like sequencing depth, if you're performing one of these studies, I recommend doing deeper sequencing, definitely better ability to detect ASD status from this. This could indicate that lower abundant species are potentially influential in autism. The variable region choice I haven't found to make a large difference, although you're going to have a lot of discrepancies. And I would say that the controls that you use in a particular study are of large importance. So unrelated siblings, you get a lot better classifiers, while related siblings they struggle, the machine learning models struggle a little bit more. So as far as breaking it down by geography, we find that classifiers that were built on data that was profiling Chinese subjects are working very, very well with about 80% accuracy, while classifiers in the US are lagging behind, which could emphasize that there's geographic differences here in autism. So finally, when we broke it down by sex, our classifiers are working better for males than females, but this could also be because females were underrepresented in the data. So with all of these kind of intermixed factors that are going on in autism, there's clearly a lot of things that we need to account for in our analyses, and we've tried to do that computationally by taking a batch correction approach. So what we'll do is we'll basically provide certain things like the variable region of a particular sample, the sex, and other factors that we've talked about, and we'll see whether this kind of improves our classifiers. And something that surprised us when we did this approach was we saw that our classifiers worked a lot better when we did a batch correction based on sex also. So this indicates that there's something unique about sex that also needs to be considered in future studies. So with all that, what are some of the conclusions and takeaways for us today? So I think that a lot of studies are trying to make generalized conclusions about the autism gut microbiome. They're relatively underpowered, and given the high complexity of this disorder with lots of different phenotypes that emerge, I think that these might be counterproductive. There's a lot of things that we need to pay careful attention to, such as what kinds of controls are being used in a particular study and how that's influencing their results, the sex of the subjects, the geographic location, the diet that might go along with different geographic cohorts, and the age is also very important to the microbiome. So we're profiling these children, and the microbiome is developing with children at different ages. So we need to be really cognizant of this when we're doing our analyses. So with that in mind, I think that trying to address this field, we need to kind of work together and publish better metadata so we can get at all of the complexity of the disease, and design longitudinal studies to capture the variability that occurs over time in the microbiome, and incorporate better techniques in the field. So all the data that I was sharing today was based on 16S data. There's a lot of limitations to that. You cannot get very good strain level identification or species level identification, and it's limited in the kind of functional perspectives that you can gain. So trying to incorporate more modalities in the analyses, as well as dietary information, and trying to provide more data from about the comorbidities that are occurring in these patients. And since we're seeing a lot of heterogeneity across different sites, I think the multi-site recruitment is really important. But I do think there's a really strong connection here that, given everything that I've said, and so I think that if we do try to work through the complexity of what's going on here, I think there's a lot of potential for diagnostics and therapeutics in the field. So our company is trying to address some of these shortcomings in a study that we're conducting with Mass General Hospital and the Lurie Center for Autism. So we are trying to recruit around 2,000 subjects with unrelated controls and sibling controls, doing 3 samples that we're going to collect from each subject. And we are going to apply a variety of different techniques to these fecal samples that we're collecting, not just 16S sequencing, metagenomic sequencing, analyzing the proteins of the microbiome, the metabolites, and getting high-resolution data. So with that, I have a lot of people to acknowledge. Big thanks to Arias, who was the project lead on this, and a big thanks to Eric, who's in the audience, who helped with a lot of the machine learning work. And we started this in collaboration with the Simons Foundation, who helped with some of the initial data gathering and processing. And a really big thanks to our scientific advisory board, Don Collin, our founder, Alessio Fasano, Andrew Goodman, and Christopher McDougall of MGH. So if you have any questions or want to collaborate on this, I encourage you to email and reach out to me. I think it's a pretty easy thing to do, potentially, to collect fecal samples if you're performing an autism study. So if you want to incorporate that and work with us, or just, I think this is a pretty promising field. So I encourage everyone to look into it. So, thank you.
Question?
Hey, how's it going? I've got a boss base coming too, so I'm going to connect afterwards. But I know it's far and wants to call some folks. I want you to get folks involved. Great presentation. So one thing I would, a few things immediately come to mind. One, I work on trying to collect stool samples for autism studies in the past, and specifically working with the American Gut Project. It's not as easy as you think. So many of these kids have chronic constipation and diarrhea. So getting a good stool sample is a little more challenging than you might anticipate. So just be prepared for that. Two, given that looking at differences between case controls, you have a lot of good ideas, but in terms of your study design that you're going to be doing prospectively, one, make sure that you guys are, looks like you have a food diary, that's good. Two, make sure that you have a really good and robust GI symptoms questionnaire. So maybe if you focus to a subgroup of autism with the GI disturbance, that might provide a little bit better signal. Three, regression, maybe something that you guys might want to look at in multiple regressions, since that's something that appears to correlate with GI symptoms. There might be a connection with microbiomes, I don't know. Those are just a few things that come to mind instantly, but I wish you the best of luck.
I know, I appreciate the comments, yeah, I definitely agree with that.
Nice presentation, thanks. One question, have you published this meta-analysis that you did on these 14?
Yeah, this is online.
Okay. So like a white paper for best practices maybe? Or suggestions about, because you said long read sequences is better than short read, and you need to do both tenders and so on, so all that's in the paper?
Yeah, I think it wasn't kind of organized as kind of a best practices, but I think if that's something that would be useful for researchers, I think that's something
Well,
that
there's a lot of discrepancy in the field, and you pointed out a lot of reasons for that. So what would really be useful is, what is the use of doing a study where you're comparing ASD versus control and looking at biome? I mean, what's the usefulness of doing that if you've done 50 studies and 14 meet the criteria and their data are all over the place? So best practices might be a good place, or recommendations might be helpful.
Yeah, and I think that's one of the reasons that we posted that online, is just to, highlight all of these factors that I think researchers need to pay attention to. And I think to answer your question of what's the future use of it, we can talk later about some of the conclusions that we've drawn from it, but we're hoping to share more about.
Oh, I misunderstood. So it's not a publication; it's just something you put on your website.
It's a preprint, yeah.
Oh, great. I'll look at it. Thank you.
Yeah.
You had a question here?
You indicated that in the ASD classification, sequence in depth had an improvement.
Yeah.
That means you must have learned from that with regards to what was in the sequence in depth, because you have enough individuals to get that correlation. So what's the bottom line?
Yeah, so I thought that was a really interesting finding, and I should note that there are a lot of potential confounders to that particular analysis. Since we only have 14 studies, it could be that the studies that happen to have more sequencing depth, they might have been from some of the Chinese cohorts that have really good classifiers also. But that being said, I do think that the increased sequencing depth is important in autism, because it's picking out more species that are at the lower end of abundance that might be missed from lower depth sequencing analyses.
And you know which ones they are?
I can look into, yeah.